Oh, we lost Danny. So we are looking at the muscular system today. Obviously, probably the system that you think of when you think of personal training. Whoops. Oh, Danny. Oh, there we go. I think you must have had two of me open. So we want to go over our final portion of the kinetic chain today and really understand the movement part of our human movement system. So, we talked about the command center that controls everything. We talked about our framework. Well, this is going to be the system that's responsible for manipulating that framework. And yes, it is under the control of that command system. So we're talking today about how it is going to adapt and how it's going to respond to exercise, which is obviously huge, considering that, this is one of the most adaptable systems in your entire body. You think about how we can train our body to do specific things. Your muscular system in particular adapts really well to stuff because it is designed to be able to grow and shrink and do all kinds of different things. Meanwhile, it's going to learn, be working with your nervous system. And so if it develops certain habits from your nervous system, that's also going to inform and affect how it operates. So your muscular system, we are going to define that as a series of muscles that are going to manipulate and move around your skeleton. Now, each individual muscle is technically its own organ. If you define organ with the classic physiologic term, which is a series of tissue that comes together. That means that technically every muscle is its own, right? If we look at the organization of how we got this. And so each of these individual little organs is going to manipulate bones around your body, right? Some muscles will just manipulate one, some will manipulate multiple. It will kind of depend on where they're attached and how they're attached and how they're generating this internal tension. So that's really the big thing, right? Your muscles are these really cool organs that generate their own internal tissue, and that internal tension. And that internal tension is actually what pulls them end to end, so they're kind of stretched out like this, and then they just pull inward like that, and that shortens the entire organ, and that is what sort of drags the bones around. So like I said, it is under the control of your nervous system. And one of the biggest things that we're going to talk about today are muscle fibers, which is just a really big fancy way of saying muscle cells. They get a special name because technically they do have all these little fibers in them that make them unique. And those little fibers are our various different proteins. And so when these proteins bind together and they move around and it manipulates things, right? It moves stuff around. So yeah, that is where we're heading. You guys know I want to start you off with a little bit of a visual aid here. So we're going to see, let's see, crash course, muscular system. So we're watching this one today. Like I said, there's going to be a lot of little tiny bits here, but I think you're going to find it fun. That is actually a pretty cool t-shirt. All right, here we go. Oh, also, really quick word of note. We love Hank. Listen to him when it comes to explanations on physiology, but never pay attention to him when it comes to exercise form, because it is always very bad. Even though we love the man. All right, here we go. Oh, good. And then, okay.
Well, hello there. You caught me while I was working out. Last time I was lifting weights during the Crash Course episode. Also, the last time I was lifting weights, we were talking about how all this is possible because of cellular respiration, the process our cells use to get and store energy from the food that we eat. Remember that? Good times. As it happens, a lot of what we learned then is also really helpful in understanding the organ system that we use to do our gun blasting and walking and fork and knife operating and parkour and playing Assassin's Creed and, moving around.
Brad, I think the volume is not good.
Oh, you're right. Thank you. Thank you. I forgot to share the sound. Okay. That button. All right, here we go.
Wouldn't be able to move them without the help of that same molecule that your cells use to get all their jobs done. Good old adenosine triphosphate. Now, your muscles may be your body's most obvious moving parts, but as with all things that are truly worth learning about, this system is both way more complex and way more awesome than it first appears. Yeah. Why? Because of chemistry. When you think of muscles, your mind usually goes straight to the guns there, but you really have three different types of muscle in your body. You have the cardiac muscle, your heart muscle, which is different from all the sorts of muscle in your body, and then you have smooth muscle, which is responsible for carrying out most of your involuntary processes like pushing food through your digestive tract and pushing blood through your arteries, important stuff there. And then there's the muscles that you're most familiar with, the skeletal muscles. Your gluteus maximus, your masseter, which is, important for chewing your hot pockets, the abductor pollicis brevis, right at the base of your thumb, aka your video game muscles, that's important for the Assassin's Creed. Just some of the 640 skeletal muscles you have. Those muscles, like all of your muscles, are only good at two things: contracting to become shorter and relaxing back out to their resting length. That's all muscles do. They contract and they relax. It's pretty amazing that you can make a ballerina out of that. If you were to peel back my skin and take a look at one of my muscles, please don't do that, but if you did, you'd see that it thickens in the middle at what's called the muscle belly, and then it tapers off on either end into a tendon. Tendons are made of fibrous proteins, mostly collagen, that connect the muscle to the bone. Just a side note, ligaments, similar to tendons, but instead they connect bones to other bones. These muscle-tendon combos stretch across one or more joints. In this case, it stretches across my elbow, so that one bone can move in relation to the other bone. So I just moved my arm, and now I'm moving my mouth, and I'm basically moving my whole body right now. And the question is, how am I doing this? How am I moving all of these things in all this sort of amazing fluid ways? How am I able to do that at all? Unfortunately, it's kind of complicated, but it's wonderful and amazing, so it will be worth it in the end. First, we need to understand the anatomy of a skeletal muscle, which includes many, many layers of long, thin strands. Think of one of your skeletal muscles as a rope. It's made of smaller ropes that are bundled together, and then those ropes are made of bundles of thread, and those threads are made of tiny, tiny filaments. This structure is what makes meat stringy, because after all, meat is just muscle. This chicken breast is, or was, the pectoralis major muscle of a chicken. It connected the bird's sternum or breastbone to the humerus in its wing, and sometimes I feel like chickens have bigger pecs than I do. This is crazy. When you peel this muscle apart, you see that it's really made up of layers of thin strings. These are muscle fascicles, and each fascicle is made up of lots and lots of smaller strands. These we can't see. They're called muscle fibers, and these are the actual muscle cells. Now, because muscle cells perform such a specialized job, they're not like your run-of-the-mill somatic cells. For starters, they each have multiple nuclei. That's because each muscle cell is actually formed by a bunch of cells, somewhat like stem cells called progenitor cells, fusing together. Muscle cells are basically just bundles of complex protein strands, and since nuclei are essential for the protein-making process, muscle cells need lots of nuclei to make all the protein they need. From here on, you'll notice, by the way, that a lot of the stuff I'm talking about starts with the prefix myo or sarco, from the Greek words for muscle or flesh, respectively. Whenever you see those terms in biology, you know you're probably in muscle country. For instance, those protein strands that I just mentioned that make up a muscle cell are called myofibrils, and each one is divided lengthwise into segments called sarcomeres. This is where the action happens, my friends, because it's the sarcomere that will actually do the contracting and relaxing to create the muscle movement. Each muscle cell has tens of thousands of these guys, and they all contract together to make you do stuff. And this contracting and relaxing occurs through this really cool and complex interaction between two different kinds of protein strands called myofilaments. One myofilament is the protein actin, which are skinny strands that attach to either one of the two ends of the sarcomere, and the other is myosin, which is thicker and studded with these little golf club-shaped knobs along it called heads. Inside a sarcomere, these proteins occur in layers, with a thick strand of myosin floating between several strands of actin. Just how many strands of actin depends on the muscle we're talking about. In this case, let's just say that there are 4, 2 sitting on top and 2 sitting on the bottom. Now, when the muscle cell is at rest, none of these strands are touching each other, but they really desperately want to. They're like middle school students at a formal dance. The myosin in particular wants nothing more than to reach its little heads up and do some heavy petting with the actin. The chemical dance that allows this to happen is one of the sexiest things that goes on in your body other than, like, sex. And it's known as the sliding filament model of muscle contraction, which reminds me, of an interesting story. I mentioned last week that we didn't really have even a passing understanding of the human skeleton until the 1500s, which seems kind of tardy to the party to me, but that's nothing compared with this. We didn't figure out how muscles worked until 1954. In 1954, two teams of researchers independently discovered that the sliding filament model is how muscles contract. And as luck would have it, two of the four scientists who made this discovery were named Huxley. We've already discussed Thomas Henry Huxley, the father of comparative anatomy and Darwin's bulldog. Well, his grandkids were all awesome at something too, like Aldous Huxley, who wrote the novel Brave New World, Julian Huxley, who was central to the development of modern evolutionary theory, and Andrew Fielding Huxley. Andrew Huxley was a physiologist who, with colleague Rolf Niedergerk, set out to solve the muscle contracting mystery. Until the early 1950s, all we knew was that myofibrils were full of protein strands. At that time, most people thought that these strands simply changed shape and shortened, like how a spring recoils after it's been stretched out. And by the 1950s, we'd learned pretty much everything we could about muscle cells by using conventional microscopes. So Huxley and Niedergerk actually designed and built a new microscope, a tricked out kind of interference microscope which uses two separate beams of light. And with that, they found that during contraction, some protein strands kept their length the same, while others around them contracted. But at the very same time, British biophysicist Gene Hansen and Hugh Esmore Huxley, an American biologist who had no relation to the famous British Huxleys, were using another newfangled tool, the electron microscope. Using that, they observed that muscle fiber was composed of thick and thin filaments, the myosin and the actin, and that the filaments were arranged in such a way that they could slide across each other to shorten the sarcomere. So in two separate papers, published the same day, in the same journal, two teams proposed that muscle contractions were caused by the movement of one protein over another. I guess an idea whose time had come. Except it's not that simple. To understand how the sliding filament model works, the first thing to keep in mind is that in addition to needing a bunch of protein, muscle cells need to make lots of ATP. ATP, you remember, creates the energy for almost everything your body does. Yes, that goes for muscle movement as well. Another thing to remember is that some proteins can change shape when they come into contact with certain ions. Like we've seen that with the sodium-potassium pumps, for instance. Those pumps are proteins that can accept sodium ions outside a cell, and then they change shape to release them inside a cell. And also, suddenly at the same time, they become able to accept potassium ions. These shape changers are how cells get a lot of the day-to-day job of living done. In a sarcomere, it's calcium ions that change the shape of some of the proteins so that the myosin can finally have its way and grope the actin strands all around it. Then it'll drag those actin strands toward each other, causing the sarcomere to contract. But when a muscle cell is at rest, there are a couple of things that keep this groping from happening. The first is a set of two proteins wrapped around the actin. They're called Tropomyosin and Troponin, and together they act as a kind of insulation. Let's just continue our middle school metaphor. They're the chaperones. They protect the actin from groping. At this point, each little head on the myosin strand has the wreckage of a spent ATP molecule stuck to it. That's ADP and a phosphate, and the energy from that broken ATP is already stored inside the head. So yeah, the myosin has a lot of pent-up frustration. Now, while the muscle cell is resting, it's preparing a stockpile of calcium ions that it will use as a trigger when it's go time. This is done by a specialized version of the smooth endoplasmic reticulum called the Sarcoplasmic Reticulum, or SR. It's wrapped around each sarcomere and it's studded with calcium pumps. These pumps are constantly burning up ATP to create a high concentration of calcium inside the SR. And of course, whenever you create a concentration gradient, you know it's going to get used. So now we're ready for a muscle contraction to start, but what starts it? Well, stimulus, of course, from a neuron. Muscles are activated by motor neurons, and each sarcomere has a motor neuron nearby. When a signal travels down the neuron to the neuron synapse with the muscle cell, it triggers the release of neurotransmitters, which in turn set off another action potential inside the muscle cell. That action potential continues along the muscle cell's membrane and then flows inside it along special folds in the membrane called T-tubules. When that signal reaches the SR inside the cell, bingo, the SR's channels open wide and let all those calcium ions diffuse down that concentration gradient. The calcium ions bind with one of the chaperones, the troponin, which causes the troponin to rotate around the actin and drag the tropomyosin out of the way, revealing all of those super hot binding sites on the actin. With our chaperones distracted, the myosin it totally goes to town. It reaches all of those little tiny heads along its length to bind up with the actin, and the excitement of that long-awaited precious contact finally releases the energy that came from breaking that ATP molecule. This burst of energy causes the heads to suddenly bend toward the center of the sarcomere, pulling the actin strands together and shrinking the sarcomere. In millions of sarcomeres, in hundreds of thousands of muscle cells, this is what allows me to, lift my arms. You wouldn't think it would be so complicated. Now, in order for the contraction to stop, you're gonna have to tear those two proteins apart, because each myosin head is really comfortable here, snuggling with its beloved actin. So it'll take another passing ATP molecule to attach to the head, which breaks off one of the phosphates to release its energy as soon as they touch. That energy breaks the myosin's bond with the actin and lowers the head, leaving it alone and frustrated once more. So it's weird that the energy from the ATP is actually used to make the muscle relax. But in fact, that's why we get rigor mortis. When you're dead, there's no more ATP to make the muscle relax, and all the calcium ions diffuse out of the sarcoplasmic reticulum, causing the muscles to enter their resting state, which is contracted. But you're not dead yet, so let's wrap this up. When the myosin and actin are being separated, the sarcoplasmic reticulum is hard at work pumping all of the calcium ions back inside it and storing them up for next time. That lets our chaperones come back. The troponin and the tropomyosin retake their positions around the actin strand, and resets the sarcomere for the next impulse to come along. Chemistry makes it all possible, from blasting your guns to my awesome dance moves. Thank you for watching this episode of Crash Course Biology. If you want to go back and look at some stuff, because it was a confusing episode today, table of contents. And thanks to everyone who helped put this together. This one was a doozy, so thanks to our head writer, Blake DiPestino. And of course, Amber, as always, for doing our amazing graphics. If you have any questions, please leave them down below in the comments, or get in touch with us on Facebook or Twitter. We will endeavor to answer you, as will all of those extraordinarily helpful people who are not affiliated with us at all, but are quite smart and helpful. So thank you to them, and we'll see you next time.
All right. So, yeah, there is a lot of little bits of anatomy, but I love that video because you can actually see all the little proteins and how they're binding and, creating this sliding action, which is, mostly what we're going to talk about today. The muscular system, like I said, it's all of those little muscles that are coming together. All those muscles are made up of individual muscle fibers or muscle cells. So a muscle is nothing more than a big compilation of these fibers arranged in bundles of bundles of bundles. Okay. So the first bundle itself, right, that's the outermost layer. That is going to be what we call your fascia. So the outermost layer, we call it fascia. It's basically like a connective tissue. Okay. And it binds everything together and keeps it from getting tangled, right? So if you look at muscle fascia, your muscle fascia is this webbing around the outside. And by the way, it has a little bit of contractile ability itself. It is connective tissue, but it sometimes can get really, really tight, and your fascia can actually cause certain aches and pains. Sometimes we see tendonitis, for instance. That's where the tendon's getting inflamed, but oftentimes it'll also pull on the fascia. If you've ever heard of plantar fasciitis, which I can never spell properly, but plantar fasciitis is nothing more than inflammation of your plantar fascia. So you can see here is your Achilles tendon, and it wraps around the heel and then becomes your plantar fascia, which is kind of muscle, but really it's just a lot of fascia. When this gets really inflamed, it rubs on this little knob here on the bottom of your calcaneus, that's your heel bone, and that rubbing sensation gets inflamed, and so it'll feel sort of a burning or a stabbing sensation in your heel. It's especially bad when you wake up in the morning, and then sort of relaxes slowly the more you move. But then if you sit down for too long, it gets inflamed again. So plantar fasciitis can be a real pain in the heel. So they're arranged with that outermost layer, of connective tissue around the outside. If we look at muscle anatomy. Oh, that's going to be all of our different muscles. Let's see here. Muscle fiber. There we go. So if you look at like what we're seeing here, you can see there's our outermost layer that I was talking about right there. And there's how it ends in a tendon like we talked about. So that's the sort of fascia. Now, the actual next outer layer, right, is going to be the outer side of the muscle itself. This is going to be what's known as your epimysium. So the epimysium is the wall that sort of surrounds the entire muscle. So we've got the we've got the outermost like fascial layer around the outside of our muscles, sort of wrapping everything. There we go. You can kind of see that whitish color a little bit better there because that's kind of what it looks like. It's a very low blood flow, not stained red tissue. But in this one, you can kind of see a little better. There's your epimysium. So the epimysium is the outermost layer of the muscle proper, right? If the fascia is its own connective tissue that surrounds the muscle, we don't consider it necessarily to be part of the muscle if we want to consider it that way. And then the next layer is going to be the epimysium, okay? And then you can see that's just bundling up together. That's holding together all of these other little bundles, right? You see this like circle here and this circle here and this circle here. Those are all like, you know, individual bundles. We call that a fascicle, okay? So let's just pull one of those out. If we pull out this fascicle here, you know, that's all a muscle is, right? A muscle is a bundling of several fascicles. In this case, it's, you know, with the one we've got pulled out here, there's this one, there's probably one behind it here. So 1, 2, 3, 4, 5, 6, 7, 8. You know, this muscle is 9 fascicles all bundled together in our little diagram here. In your real muscles, it would be very, very different from that. But this is sort of what it looks like, right? So a muscle is a bundle of fascicles. Now, a fascicle has its own connective tissue. It's actually got its own tissue that is sort of wrapping around its outermost layer. That outermost layer right there of the fascicle, we are going to call that the paramecium, okay? So the paramecium is around the perimeter of the fascicle, and it serves as like a way to kind of wrap everything together. So imagine if you were like building a muscle, right? Or let's just say you're you know, this is gonna be a weird example, but let's just say you're building a raft, right? Like you're stranded on a deserted island and you need to build a raft. You got plenty of rope and you got plenty of sticks. You know, those are the tools you have access to, right? You need to be able to like bundle all of this together, right? In order to like get it to stay. And so like that rope is going to be used to like hold these bundles, right? That's what we're seeing. We're just seeing different layers of that, right? So what is a fascicle? Well, a fascicle is a bundling, a wrapping of several muscle cells all coming together. And that's what we're seeing here, right? This is actually an individual muscle fiber, okay? So we can see here's our individual muscle fiber. That muscle fiber is wrapped up with a bunch of other muscle fibers. In the picture we're looking at now, it's 1, 2, 3, 4, 5, 6, 7. It's also 9. Again, that's a coincidence. This is not how the anatomy actually works, but it makes a good diagram. So you can see like these 9 muscle cells, this one and all the others, are all bound together in a bundle, and we call that bundle a fascicle. And then if that fascicle is tied together with a whole bunch of other fascicles, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, right? There's the actual, you know, that's what makes up the muscle itself. So, and then all of it's going to be wrapped in connective tissue called fascia. So the outermost layer is just going to be a connective tissue. That's our fascia. The next layer is going to be the outermost layer of the muscle. And then the next layer coming inward is going to be the wrapper around a fascicle. And then there's one final layer around the outside cell wall of every individual muscle fiber. That's going to be called your endomysium. And the endomysium is basically just like the wall that separates one cell from another. You know, it's like the outer, but like every one of your cells has like a wall around it to keep fluid in and fluid out. And that's what we're seeing here, right? So you can see this little, like, it's kind of hard to see probably, but it's a little white, you know, wall there. That's the wrapper around the outside of the muscle cell. Now, you can see there's another little strand here being pulled out of this muscle cell. And that's what's called a myofibril. Okay, and we're going to look at that here in just a little bit. But a myofibril is this big long rod. It's a special structure found only in muscle cells. That rod is what allows muscle cells to contract. And so if you consider that like this rod is contracting, meanwhile all the myofibrils in that muscle cell are contracting, that's going to cause the entire muscle cell to shorten. And then all the other muscle cells are going to shorten at the same time inside that fascicle. And so the whole fascicle is going to get shorter. And then all the other fascicles are doing the exact same thing at the exact same time. So the entire muscle is going to get shorter. And when that happens, that's what manipulates and moves our bones around. It's also why like our muscles kind of bunch up when we contract them, you know? So that's the different layers that we're going to see. The outermost fascia wraps the muscle itself. The muscle wraps all of the fascicles. That's called the epimysium. And then the fascicle is going to wrap itself. That's going to hold all the muscle fibers, muscle cells, whatever you want to call it. That's the paramecium. And then each individual muscle cell has a cell wall called the endomysium. So bundles of, again, it's a bundle of bundles all wrapped together in various different layers. And like I said, all of that is going to end in connective tissue that we call a tendon. Okay, so that tendon is the connective tissue that actually inserts onto the bone, right? I always like to use the analogy, we talked about how our bones are very holy, right? We talked about how like our bones are porous, right? They've got it's sort of like a honeycomb shape. When you look at bone tissue, it looks something kind of like this, right? And I always like to think of like your tendon, honestly, because it does sort of look like this. Think of it as like hot glue, right? If you've ever used a hot glue gun at any point in your life, or even just regular glue, the way it works is like, you heat that stuff up. It's almost like plastic or rubber, and you lay it onto something. If you used hot glue gun on like, I don't know, like a sheet of metal, it would probably get a little bit of grip, but it's not going to get like the most stellar grip in the world because it's a very smooth tissue. It's not as porous, right? But if you were to like take a hot glue gun and you threaded it into like this bone tissue here, it would get in between all the little holes and it would get a really good grip on things. And that's how your tendons actually insert. It's connective tissue that kind of feeds in and gets into all these holes. And so it gets a really good grip on the bones. And then all of your muscle cells are coming to the end, and all that fiber and connective tissue that's also getting kind of wrapped around and tangled up with that tendon as well. And so that's how all of this is sort of tying together. And then it has contractile elements to it, similar to how our muscles do, and that's why your tendons are part of the muscle, but it's also sort of inserting onto the bone. And so again, this is that love-hate picture that I have from yesterday. When we were looking at the skeletal system, you can see like a ligament here, which is also very similar to a tendon. You can see this is a ligament attaching a bone to a bone. And then also in this picture, you can see here is a tendon attaching a muscle to a bone. Now, here's what I love and hate about this image here. This kind of makes it look like your quadriceps tendon inserts onto the top of your kneecap, your patella, and then your patellar tendon originates from the bottom of the kneecap and then inserts onto the tibia. That's not quite how this works, actually. It's actually embeds right over top of it. Yes, some of it does grab the top and some of it grabs the bottom, and that's how it actually moves your kneecap around. But it actually runs over top of this. So imagine kind of shading in this over top. That's a little bit more scientifically accurate. In fact, that's what's kind of funny. If you touch your kneecap, you're actually pinching between your finger and the bone itself, your tendon there, and so a little bit of soft tissue, which is why it feels a little weird if you ever rub just your kneecap itself. So we want to make sure that that is being able, moving the way it's supposed to, because this kneecap needs to be able to kind of slide up this way and then fall back into place. But anyway, the other thing that's confusing is like these tendons and ligaments look the same in this diagram, which is another thing that I love and hate about this picture. The reason we're drawing them both in white, the reason they look very similar, they both get very little blood flow. And so this is where we run into most of our injury problems, right? Musculoskeletal problems are often caused from like tendons and ligaments. Those are the areas that we're often most struggling with. So that's what a tendon is. Like I said, they attach your muscles to your bones. They have limited blood flow and they repair and they adapt much more slowly. All right. Questions, comments, concerns. I know the layers thing is a little bit confusing. Honestly, we don't talk about the layers that much, guys. You really just need to know like a muscle is a bunch of fascicles and a fascicle is a bunch of muscle cells wrapped together. So you have a lot of individual fascicles inside of each muscle. Hold on. Danny's having technical troubles. Let me text him individually. Try unplugging it and plugging it back in. Oh, he's back. Never mind. Danny, I was just getting ready to text you. Oh wait, there's his audio. There we go. Now you're back. How you doing?
Is my internet or
I think it has to. I don't know. I would assume so. But sometimes like there are days where like I'll be kicking people out left and right, and I'm like, is it me? Is it like the host of the call? I don't know.
Okay.
I'm not sure. Oh, for instance, we just lost Cyrus too. Oh, and now we got two Cyruses. Okay, there we go. Hey, welcome back, Cyrus.
Yes, sir. I don't know why you just kicked me out.
I don't know. I might be kicking people out left and right. Zoom's like, get on with the lesson, man. Let's go. Like,
It's Friday. Says it's Friday. Let's go.
Yeah, that's what it is. My Zoom's
you,
like, hey man, I got a beach appointment. We got to wrap this. Or I'm being hacked. No. All right, so let's go ahead and look at individual muscle fibers here. Let's take a look at some of this microanatomy, right? So we did talk about, all the proteins in the video, right? We saw all those individual little proteins that are allowing these contractions to take place. So for the record, like your muscle cells are just like any other cell inside your body. Like if you look at like an animal cell, we always draw it in biology. We draw it like this, right? Like this is, you guys remember like high school biology. This is every cell you ever looked at, and it's like, this is the nucleus, this is the brain of the cell. And then, here's the little mitochondria, that's the powerhouse of the cell. And I said that, every high school teacher says that. For the record, this is what's kind of funny. None of your cells look like this. None of them. If they do, because here's the thing, this cell has no function. It doesn't do anything special because it's just a blob that this looks like an amoeba. It could maybe wiggle around a little bit and maybe eat something, but for the most part, the cell can't do anything. All of your cells in your actual body have this plus something that makes them special. So something that looks a little bit different. So for instance, like an adipocyte cell would look like this. It would look just like the other cell, right? It would have all the normal stuff. And you can kind of see it there, right? There's the all the normal stuff in it. But see this like big sac in the center? That special sac is what makes that cell actually do something. In this case, it's a fat cell. This big sac stores triglycerides, and that's what our fat cells look like. So none of your cells actually look like the classic high school biology cell. They all have all of that plus something special. So like a neuron, for instance, neuron organelles. Hopefully that gives me the picture I want. Yeah, there you go. There's what a neuron looks like, right? Remember we talked about how it has all the like little fingers? There's all the that's the classic animal cell plus all this other stuff. So that's the thing here that we need to remember. So a muscle fiber gets a special name because it's a muscle cell. It has all that normal stuff in it, plus a bunch of other stuff. And that other stuff is what we're going to talk about. That's what I think is really exciting. So, so for instance, like inside of a normal animal cell, we have fluid, right? All of your cells have fluid. You're mostly made up of water, you know, and that fluid is called cytoplasm. Cytoplasm is the name for the fluid when it's got a suspension of stuff in it, right? Electrolytes and proteins and organelles, you know, all the stuff flowing around in the fluid itself. The fluid is called cytosol. The fluid, when it has stuff in it, it's called cytoplasm. So we're just going to call it cytoplasm because it always has stuff in it. But in a muscle cell, we're going to call it sarcoplasm. Right? Sarcoplasm is the liquid portion of your muscle cell. And so it contains all this special stuff in it related directly to our muscles, right? So it's going to have like glycogen, so it can break it down and produce energy out of carbohydrates. It's going to have fat, so it can break it down and produce energy out of fats. It's going to have minerals to allow this, you know, electricity to get created for the cell to kind of power itself. And then it's going to have like what's called myoglobin, which is a protein that binds to oxygen and moves oxygen from place to place, because everything I just talked about takes oxygen, requires that, right? So the sarcoplasm is the fluid portion of a muscle cell. Sarco coming from the prefix that means flesh, plasm coming from fluid. So it's flesh fluid. Don't think too much about it, which is really just a protein drink, right? That's all protein shakes are. They're just flesh fluid. No, I'm kidding. Inside your muscle cell, you're also going to find nuclei, which is the brain of your cell. What's kind of fun about muscle cells, guys, is they actually have lots of nuclei. Remember, your nucleus of a cell, it's the brain of the cell. It contains all your DNA. So muscle cells actually have lots of nuclei. And the reason for that is because muscle cells need to be able to grow really quickly. So they need to be able to assemble DNA and assemble proteins really, really fast. So by having lots of actual nuclei, it makes it very easy for the muscle to grow and develop because the nuclei can just sort of transform into like little proteins and stuff. So that's those are called progenitor cells. Really, really cool stuff. So that's the nuclei. You've also got mitochondria. Mitochondria are going to produce energy for your muscles. Obviously, that's a really big thing. We want to make sure that our muscles are continually producing energy, because if they can do that, they can actually contract. But probably the coolest thing, and then there's another example of what we're seeing there, probably the coolest thing inside of your muscle cells are going to be what are called your myofibrils. So myofibrils are very special within muscle cells themselves. And they're these big, long rods, okay? They're these big, long rods. And on those rods live all of these individual little proteins. And those proteins are where the actual contraction takes place. And remember, we're in one muscle cell right now. So a muscle cell is going to have a whole bunch of what are called myofibrils, okay? And a myofibril is this big, long rod that just lives within your muscles, right? So I'm going to go ahead and just open this image here. Let's see, open this in a new tab, and then I'm going to hate it when it's one of these. Copy image. We'll go over to Paint here. Okay, so here's the individual like myofibril, right? So calculator, Paint. That's the individual myofibril there, right? Now, in an actual muscle, it would look, you know, probably like this, actually. It would just be, you know, tied together, and like, you know, it would be a lot longer than this, running through the entire muscle from end to end, right? Tied together like that. And then here's the thing, it would also, there would be a whole, oh shoot, no, I didn't. There would be a whole bunch of these myofibrils all tied together. So there would just be like an actual muscle would just look something kind of like this. Remember, this is in one individual muscle cell. So there are just tons and tons and tons of these rods packed into your muscle cell, right? So we want to understand sort of what's going on in this rod. So just let's zoom in just on this one individual myofibril here. Let's go ahead and delete this. And I'll just zoom in here. So there's an individual myofibril. Like, well, actually, you know what? I want one. Hold on. I want one myofibril. Okay, so there's our one myofibril. Look at this, guys. You can see, here's the thing about it, right? Notice how there are these like thick, these little bands in the middle. That's called your A band. But then see this like little zigzag action here? That is called your Z line. Okay. And then here's another Z line over here, right? And what that does is the way these proteins are arranged, they're just slightly offset. And that gives it a little bit more stability, right? Like they're stacking kind of like when you stack like bricks in a house, right? You notice how like you don't just take like if you're building like a brick wall, you don't just like, you know, build the bricks in one straight line and like another straight line, right? You offset them slightly. Like you put this row here and then this one, you know, it doesn't stack perfectly, it stacks off to the side so that they're just getting like half and all this extra contact. That's actually how your myofibrils are arranged. So end to end, and you can just see that all stacked together. Like, look, see how all those myofibrils are all bound together, right? That's one muscle cell. And then that muscle cell is going to be bound with another muscle cell and another muscle cell, and all those muscle cells are going to be bound together. That's a fascicle. And then that fascicle is bound together with a bunch of other fascicles. That's the muscle. Bundles of bundles of bundles of bundles of bundles. You know, like it's crazy how much of this there is in our muscles, right? But here's the thing about like that zigzag action. While it gives it a lot of stability, right? And so the area between that zigzagging is actually where the shortening is going to take place. So what's actually going to happen here is if we look at this little zigzag here, you're going to see there is going to be contraction that is going to move it this way. And then there's going to be contraction that moves it this way. And meanwhile, the exact same thing is happening along the entire myofibril, right? So it's happening over here and it's going to happen over here, right? And so the entire cell, it's just like pulling like this. And what that's going to do, the entire rod is going to shorten in kind of like this, right? It's going to pull from end to end, and that's going to happen in all of those rods simultaneously. So the myofibrils are the rods, okay? And then the reason they're contracting is because of proteins that live within the rods, okay? So actin is this thin little filament. You can actually see it right here. If we zoom in just a little bit, this is actin. So there's that band, right? There's the band. And then see how there's this like thick bit in the middle. And then there's these like little white lines. This is those white lines. This is the thin little strand right here. This is a molecule of what we call actin, right? So I got a picture of it here in the PowerPoint as well. Looks a little bit different. And you can see if we zoom in, right? They turn this on its end here, but you can see these little thin strands. Those are your actin strands. And then I don't know why they did this, but they drew it with a straight line here. You know, if you actually look at this, see, there's the zigzag, right? That's the Z line we were talking about. So actin is this thin little strand, right? And then there's a gap. And then there's more little actin strands here, right? So actin is this thin little protein, and actin has all these little binding sites on it, right? All these little tiny itty bitty binding sites. And those binding sites are very attractive to this other protein in the middle. This thing, doesn't this look like kind of like a Dr. Seuss tree to you guys? Right? Like these little weird little club, like this little branch here, and it's got all these little branches on it, right? Like to me, that looks like something right out of a Dr. Seuss book, you know, like a weird Dr. Seuss tree. But all those little heads, all those little like club little branches that are coming off of there, they are super, super attracted to actin's binding sites. So they want to grab actin's binding sites all the time, right? And that's what we're going to see. So what's going to happen is, like, let's say like I'm dead center, right? Like let's say I'm like right here along like the A band, right? And you know, I've got these little club-like heads right here on both sides. What's going to happen is the club-like heads are going to reach up here and they're going to grab that binding site, right? They're going to reach up and grab over here. And then this one's going to reach up and grab over here. And what's going to happen is I'm going to pull towards my center like this. And that's what's going to shorten this entire little section between this Z line and that Z line. Okay. And so the entire Z line is going to move this way. That's what we're seeing here, right? So you can see, right, from the middle, these little club-like heads. That's called myosin, by the way. The thick tree in the middle, that's our myosin, right? And then the thin little white strands, those are called actin. And so myosin is going to reach up over here, and it's going to reach up over here, and it's going to pull in towards the center. And the entire sarcomere, which is this area here, right, between these two, this little box here, this is a sarcomere, right? The entire sarcomere is going to shorten. Meanwhile, this sarcomere is also going to shorten, and that sarcomere is going to shorten. So don't think of a sarcomere is not like a physical thing that exists. Think of it as like an area, right? It's an area along the myofibril. The myofibril is this rod, and it's a thing you can hold, right? The myofibril is the rod, and it's got all these different sections of sarcomeres, right? So that is what we're going to see there, right? So all of these individual fibers. So then you can see here's a contracted muscle versus a relaxed muscle here, right? A contracted muscle is where all the binding sites have slid the actin towards the center, right? It pulled this actin, this way, and it pulled this actin this way. And so the whole strand is really short and contracted. Meanwhile, a relaxed muscle, look at how much space there is, right? It's got all this room to slide this way and all this room to slide this way, and all those little club-like heads. So let's see this in sort of an animation here. Okay, so we can understand this a little bit better. So there you can actually see, right? Look at all these sarcomeres, right? This is like this is a sarcomere, and that's these are all individual, like this is all taking place within one freaking myofibril, and your muscle cells are made up of thousands of myofibrils, right? And you can see like the whole thing is just sliding towards the center there, right? And so they're just grabbing those little heads and pulling straight in towards the middle, right? I don't love that animation actually. And look, I love this one. Like, look at how freaking complicated this picture is, right? Like, just a bajillion of them. Like, I don't know who rendered that. Like, that computer must have taken forever to do that. But here's how it's actually happening, right? There's the little heads on the end of myosin, and there's all the little binding sites. Those little blue things, those are the binding sites. So the heads reach up and they pull. And so they pull towards the center here, and they pull towards the center here, and the entire thing shortens. Now, that process of shortening, this entire, all these proteins pulling towards the middle, we are going to call that the sliding filament theory, or the sliding filament model, if you want to call it that. I always learned it as the sliding filament theory, but I think they call it the sliding filament model nowadays. And that is where this sliding action is happening, right? Because you're seeing, right, if we look at these contractions, is the entire sarcomere is getting shorter. It really is actually like sliding, right? See that? There we go. That's actually a great picture of the slide, right? You can see the sliding action occurring, right? It's just pulling towards its center. So now we start to understand the difference between a contracted muscle and a relaxed muscle is, are the proteins all bound together? That's a contracted muscle. Or are the proteins all relaxed? Now, all of this is taking place because your nervous system is telling it to happen, right? It's actually making it happen by flushing different materials into the area that causes this binding to occur. Because here's the thing, I just talked about how like these fibers are super attracted to one another. They want to be bound together. They want to be contracted. They want to be in this shortened up, like contracted state 24 hours a day, 365 days a year, right? They are Romeo and Juliet, or they're teenagers at a high school dance who just want to be all up in each other's business, but we need to be able to relax our muscles. We need to be able to get in there and push those proteins apart. So luckily for us, we actually have another protein that actually lives on actin. And so this protein, these proteins, I should say, are called troponin and tropomyosin. Troponin and tropomyosin are regulatory proteins that block the binding from happening. They keep the contraction from occurring so that we can relax our muscles, okay? So they get in there and they basically pull this apart, okay? And so you can actually see that here, right? This is how a contraction takes place. Like right now, the muscle's relaxed, right? It's relaxed now, but then see this little green strip here and this like yellow band there, right? They're getting moved out of the way. When they move out of the way, that's where the binding sites are exposed. So then myosin gropes the actin and it pulls towards the center. So when we contract, when we relax our muscles, we just put those chaperones, troponin and tropomyosin, we put them back into place. And that allows the relaxation to occur, right? So let's see if I can find a good picture of that. Troponin and tropo. Oh, that's tropomyosis. So there's sort of what that actually looks like. You can see this like band here that's kind of blocking those binding sites, right? But here's sort of a GIF of what it looks like when they're moved out of the way. So they move out of the way and they expose the binding sites, and so this actually reaches up and grabs, right? So that's what's happening inside of our muscles, right? So if we talk about this process from start to finish, right? If we go through like a muscle contraction where, you know, one sarcomere is gonna shorten, right, and pull towards its center, right? You can kind of see in the PowerPoint here, we got a really good diagram of this. This is a picture directly from your Trail Guide to Movement, by the way. You can see like how this is gonna look, right? So this is a relaxed muscle, okay? This relaxed muscle is relaxed because troponin and tropomyosin are currently blocking all the binding sites. So then your nervous system is going to zap your muscle. Okay, it's going to say, hey, I need you to contract. So what's going to happen is your nervous system is actually going to trigger the release of, and this is going to sound kind of weird, calcium. Calcium is actually what troponin and tropomyosin are super attracted to. Like actin and myosin are attracted to one another. Troponin and tropomyosin, they block the binding sites, but they're super attracted to calcium. So when calcium is in the area, it actually causes the muscle to contract. This is actually what's kind of funny. If you were to inject liquid calcium into like a muscle, it would actually allow those muscle fibers would actually contract, which is kind of funny. That's why one of the reasons, this is one of the reasons why if you've ever experienced a muscle spasm or really bad cramping, one of the things doctors will actually test when they test your blood is they're going to look at your blood calcium levels to make sure that you are actually getting enough calcium in your diet to be able to contract your muscles properly, because that could explain why you're experiencing your spasm. So we're going to look at this contraction, right? So your nervous system, your neuron zaps a muscle fiber that causes the release of calcium into the sarcomere, okay, the area between these two Z lines with all the overlapping proteins. Now that calcium is in there, troponin and tropomyosin, they're going to move away, and that they're like, all right, well, I don't care, I'm going over here, you know, like, and so they move out of the way, and now the binding sites are exposed. So myosin with its little heads are going to reach up. You know, we got a relaxed muscle here. They're going to reach up and they're going to grab actin's binding sites, and they're going to kind of pull hand over fist. It's going to grab that binding site. It's going to move this way. Next one's going to grab that binding site. Meanwhile, this one's relaxing and grabbing the next one, right? And it's just kind of pulling hand over fist. And the entire muscle cell or the entire sarcomere is shortening, right? So it's sliding. And now you can see there's very little space here and there's very little space here. This is a fully contracted muscle. So it went from being this long to being this long, right? Because of this sliding action. So all the proteins are sliding across one another, right? And that's really where all of this is occurring, right? That's sort of where all those proteins are actually doing all of this. Now, eventually, all that calcium will get flushed out of the area. When the nervous system signal relaxes and stops sending that actually flushes calcium out of the area. Now that there's no more calcium, troponin and tropomyosin are like, all right, let's get back to the high schoolers. We can only imagine what they've been doing, right? They're like the chaperones who get in there and like, and they push all of them apart. It's like, we said 6 inches at the dance, you know, like that's, you know, they're like, you gotta know, your parents are gonna be furious. You guys remember high school, you know, like, and so that's sort of like where the neural activation, you know, ends, all the calcium's flushed out, the chaperones go back into place, they push the proteins apart, and the muscle relaxes, getting ready for its next contraction. So all of this is under the control of your nervous system. Your nervous system is going to use what we call neural activation to get all of this to happen, right? So neural activation is the contraction of a muscle by neural stimulation. So what we'll see is like what's called a motor unit, okay? So a motor unit is one neuron, and then that neuron's going to have all these little fingers that branch out from it, and it's going to zap a whole bunch of individual muscle fibers. So remember, a muscle fiber is one muscle cell, and it's got all these myofibrils in it and thousands upon thousands of actin and myosin proteins, okay? But the nervous system is zapping one cell. So one thing that we need to remember is what's called the all or nothing principle. And it's this principle that states that when a muscle cell contracts, it will always contract absolutely as hard as it can every single time. So when you hear that, you might be thinking, well, that can't be true because, you know, I can lift this glass of water and I'm not just like slamming it into my face, you know, like I'm not contracting as hard as I can every single time. So what the heck, right? Well, remember, I said a muscle fiber will contract as hard as it can every single time, one individual muscle cell. So one of the ways that we control our strength is we use a different number of muscle fibers. Now, one individual neuron might be attached to very few muscle fibers, or it might be attached to a whole bunch of muscle fibers. Or maybe you want to create like a major super intense contraction. So you know what you're going to do? You're going to recruit multiple neurons, which will recruit multiple muscle fibers. So for instance, you can see what we're talking about here, right? This is actually the nervous system sort of threaded through our muscle cell or our muscle here, right? So this is a nerve that's going to branch out, and then it's going to zap 1, 2, 3, 4 muscle fibers, right? So maybe I use 4 muscle fibers, right? 4 individual muscle cells are going to contract. That's not a very strong contraction. So maybe that's like, you know, me using a fork while I'm like eating dinner. But what if I did like another neuron that came down and zapped 4 more muscle fibers, and another neuron that came down and zapped 4 more muscle fibers? So now, you know, I went from 4 to 8 to 12. I'm contracting a lot of muscle fibers now, right? That's a big motor unit, right? Well, maybe that's what allows me to lift a dumbbell. So this is one of the ways that our body controls our strength. So we really are, when we're in the gym and we're training, right, we are trying to create more muscle fibers, but we are also trying to create a nervous system that can control all those muscle fibers, which is why strength is not just about muscle, it's about your nervous system being able to control your muscles, right? So a motor unit is a motor neuron and all of the fibers that it innervates, you know? So motor neurons are attached to these muscle fibers. And when that neuron contracts, it will innervate all of the muscle fibers that it's attached to contract fully. Okay, that's the all or nothing principle that we'll see in just a second. It doesn't have any control over it. The way it controls strength is it just recruits a certain number. And actually, if you look at it, there's a diagram here. It kind of explains the all or nothing principle, right? So there we go. So 1 neuron for 3, 2 neurons for 6, 3 neurons for 9. Or maybe this is 1 neuron. This is Bob, and Bob only recruits 3 muscle fibers, and this is John, and John recruits 6, and this is Sally, she recruits 9, right? And so I haven't heard any of those names in a while, except for John. John's around all the time. But like that's, you know, this is a strong contraction. You would use that neuron, right? This is a weak contraction. You would use this neuron, right? So that's actually how we control our strength. You know, so when you go to the gym and you try to pick up like a big heavy dumbbell, oh, there's a really good diagram as well. You can actually see, you know, here's the difference between, you know, 2 plates or 4 plates and I guess that's 8 plates. It's a big jump there. But yeah, that's the difference between like how many neurons you're sort of recruiting to do this. Now, like I said, your neuron has to be able to do this and zap those muscle fibers. It's going to use a specialized chemical to do that. That chemical is called acetylcholine. Acetylcholine is a neurotransmitter. And so there's actually a little bit of a gap between the neuron and the muscle fiber. That's called the synaptic cleft. You don't need to necessarily know too much about this, but basically there's like the little gap, right? There's a little gap between the muscle. I think this is a neuron to another neuron in this diagram, but it works exactly the same. Let's see here. Neuromuscular junction is the other name for it. There we go. So you can see like there's a little gap in between there's the actin and myosin. See those proteins there, but there's the neuron, right? And there's this little gap here. So your neuron is going to use electricity. The electricity is going to run down, and then it's going to release these little purple dots here. Those have this little chemical called acetylcholine in them. And so they're going to spit acetylcholine out, and it's going to bind to your muscle. And when that happens, that triggers the release of all the calcium I talked about earlier. So acetylcholine binding to the muscle fiber is what causes calcium to get flushed into the area. And so those calcium ions then move troponin and tropomyosin out of the way, and all of a sudden the binding can happen. And that's where the sliding and the sliding filament actually occurs, right? So again, looking at our all or nothing principle here, right? Regardless of the number of individual muscle fibers, when a motor unit contracts, it will always contract as hard as it can. The difference between a strong contraction and a weak contraction is how many motor units are being recruited, okay? That is really what we mean when we talk about this sliding filament theory, right? You can see the muscle is lengthened, and look at how much space the muscle is lengthened. Look at how much space there is to contract, right? This is a relaxed muscle. But then as it bunches up, that space diminishes. Now that's really great. You know, that's really great when we want to do this. But, and by the way, here's the entire process I just talked about laid out in like a giant paragraph. I don't need you guys to read this slide. It's in there in case you want to like fully understand this, but this is basically just start to finish. What it's saying is like a nerve impulse travels from neuron to neuron to neuron to neuron until eventually it gets to a muscle, and then it releases acetylcholine. Acetylcholine flushes calcium into the muscle cell. Calcium moves the regulatory proteins that are troponin and tropomyosin out of the way. Actin binds to myosin, and the entire sarcomere shortens. Big freaking paragraph. And you can see that kind of happening here actually, right? The proteins move out of the way, this binds and it slides, and then eventually it relaxes, and the process can just repeat itself over and over and over again. So the all or nothing principle, right? The difference between a strong contraction and a weak contraction is how many muscle fibers are going to get recruited. And that's why it's very important for us to have lots of muscle fibers if we want to be strong. Yes, there is a muscle fiber number aspect to this contraction thing. But the other difference is we also need to make sure that we have a nervous system that knows how to control those muscle fibers. So again, when you're training for strength, you're not just training to make your muscles bigger, you are also training your nervous system, which is why we take such long rest periods in strength training, right? If I'm trying to gain muscle size, I don't need to take a lot of rest period. I just need to create a lot of like cellular damage so that my body makes it bigger. And it's like, oh man, I got all beat up. Let's make it bigger, right? But if I want to get strong, I need to take a lot of rest so that I can lift really heavy every single time. By taking a lot of rest, I get to lift really, really, really heavy. By lifting really, really heavy, I'm using my entire nervous system. And so then my nervous system, this is why anybody in here, if you've ever done really true, like maximal strength lifting, you leave the gym feeling like physically exhausted, but you also feel a little mentally exhausted. And that's your nervous system is actually worn out. You'll see this sometimes where people will literally just, it looks like they're losing their mind in the middle of a lift, and their muscles are contracting. Nervous system failure, barbell squat. Didn't find a video of this. Stretch fatigue. Trying to find a good example here. I've seen a million videos of this.
Hey guys, Greg Knuckles here.
Oh, it's Greg Knuckles. This guy's a friggin' genius. Although he faked me out with a thumbnail that doesn't reflect his his video. Love Greg Knuckles though. If you ever get a chance to watch that guy's videos, he's great. Anyway, sometimes you'll see like nervous system failure in the gym. You'll see people like, and like they're getting to the end of their lift and they're done, but you'll see them like wiggle, and like it's because their nervous system is just like, what do we do? I don't know, contract stuff, and it literally is just so exhausted. It's just firing like crazy. So that's one of the reasons why I want you to understand the all or nothing principle. So when we look at like a precise movement, right, like really tiny, not a hard contraction, it might only take 10 to 20 muscle fibers to get that, you know, like moving your eyeballs, right? 10 to 20 muscle fibers or moving my eyeballs around. But we look at like large muscles. Let's take a look at like just your gastrocnemius, which isn't actually that large of a muscle. Your calf muscles aren't that big. Not trying to trigger anybody. I know it's a sensitive topic for some. But if you look at like a larger muscle, like the gastrocnemius, your calf, right? You might be using 2,000 to 3,000. Imagine how many you're using in your gluteus maximus. That's the biggest muscle in your body, right? Tens of thousands. So those are our various muscle fiber types, or those are our various like ways that muscle fibers contract. It's very, very complicated. I know there's a lot of working parts, but the reason I want you to understand this is because like if you want to make a muscle bigger, you're going to make more proteins. If you want to make a muscle stronger, you're going to make more proteins, and you're going to make more neurons that are going to control those proteins. That's why we're looking at this. The other reason why is because we got to understand that there is a sliding action that occurs here. Now, your clients are going to have altered posture, right? Let's look at static postural distortions. So if we look at someone who's got a static postural distortion, let's say they stand like this, right? They stand with like pronate. Actually, you know what? Let's use a different example. We'll use, let's say a scoliosis. Actually, that's a great example. Let's say we got a client who's experiencing a scoliosis, and they weren't born with it. They weren't born with a scoliosis, but they have one now. We call that a functional scoliosis. If you have a structural scoliosis, you're kind of stuck with it. It's how you were born, right? Surgery can fix it. There are little things you can do, but it's a deformity, right? Functional scoliosis is somebody who developed a scoliosis because maybe they just sit on their leg all day, or they got a wall, they sit on their wallet all day, or maybe they carry something on one shoulder every time. See it in firefighters all the time, by the way, because they're always carrying like stuff the same exact way. See it in furniture movers all the time. So they got they have a scoliosis, but they weren't born with it. So you can see here sort of what we mean, right? So she's got a scoliosis. You can see, look at the gap between her arms. See how thin the gap is here versus how wide it is here? That's actually showing us that her spine is a little rotated. And then you can even see, if you looked even more carefully, you can see like her feet aren't lining up properly. There's a little, like, look at how sharp that angle is there compared to how like short it is there, right? Now, maybe she was born with this, or, maybe she's developed it over time, but either way, her pelvis isn't sitting level. So then her spine has to tilt back the other way in order to get her level back at the top, right? So because this kicks out this way, because this kicks out this way, this has to kick out this way. So there are muscles in the back. There's some muscle right here that runs from the pelvis up to your bottom rib, pelvis to bottom rib, called your quadratus lumborum. Looks like this, okay? Now imagine if these muscle fibers here, right? Imagine if some of these muscle fibers were really stretched out, right? Remember how we talked about like the actin and myosin proteins, right? When this muscle contracts, what does it do? It pulls down, some of it pulls that way and the other part of it pulls that way, right? And so the whole muscle shortens towards its center, right? Because all the myofibrils. But imagine if we took this person and we really just super hard contracted, towards the center here, and super hard contracted towards the center here, the entire muscle would be really short. And now, because this muscle's on this side, this muscle over here, it's going to get lengthened out. So maybe it's only contracting, or maybe it's even being pulled apart, being pulled that way and being pulled that way. So now what you have is what's called a muscle imbalance. Overactive or underactive muscles. So one muscle on one side of the joint's pulling too much, and the other muscle on the other side of the joint is not pulling enough. So you got this big fat contracted muscle over here, and this long, skinny, weak, relaxed muscle over here. And so it's pulling your bones in this direction, right? That would cause your spine to tilt like this. So we can see here's a relaxed QL over here and a contracted QL over here. So if I did that, you know what it would do? It would raise my hip up like this. And then I would stand, right? I'm gonna raise my hip up and I'm gonna plant my feet back down to the ground. And now look, I have got a scoliosis, right? So now I'm tilting this way. So what do I do if I wanna stand straight? I tilt back that way. And now look at the gap on my arms. You can kind of see there's a bigger gap over here and a complete lack of gap over here, which is exactly what we're seeing in her body right there, right? That is overactivity. Let's look at an example that doesn't look so extreme and might just be like, a person just standing during the day. What if somebody was standing, just kind of like I'm standing right now? Doesn't look too bad, right? But do you see how I'm like rounding my shoulders? I should be standing fully upright and pulling my shoulders back, but I've got this rounded posture here, right? That rounded posture is muscles in the front of my body contracting and staying contracted, and muscles on the back of my body being pulled apart, relaxing. So we're seeing some of them are slid and some of them are pulled apart. And that's the overactive, underactive muscle thing that we are going to be looking at for this whole class. I know that sometimes it's like microanatomy stuff, it gets really technical, but it does have direct implications. Like when I look at someone and I see, oh, their posture's messed up, I know that one muscle is short and needs to be pulled apart, and the other muscle is, is lengthened. It needs to be contracted back to neutral, and that's why we analyze posture and how we use this to happen. So I would just contract those weak fibers until they get nice and strong. And then I would stretch out those contracted fibers until they get nice and mobile. Now, that was a big section. I've been talking for an hour without giving you guys time to chime in. Does anybody have any questions? I know that was a really big chunk, but any questions or comments? How are you guys feeling? Do you feel like you got this a little bit?
Yeah, I feel like I got it a little bit. It's a lot, but I got it a little bit.
Yeah. So here's the thing. For Marjan and George, this is going to come up again. I promise. We do it every mod. We review it every mod. Next mod, I'm probably going to spend a lot of time talking about the same stuff I talked about this time. Mods after that, we'll talk a little bit more about that chemistry. We'll look more at the neurology parts. There's sort of a different way to look at it, but we will revisit it at least 4 times, and then Mo will do it twice.
That's good.
Yeah.
Yeah, cool. Because there's a lot.
I know, right?
It's a lot. Oh
but
my God.
Danny, I've had you for 2 mods now, right? Do you feel a little better than last time? Maybe not. You don't have to have full understanding yet, but do you feel better?
Yeah.
Right? It comes back around.
Uh-huh.
It's just like, is that like we need to know everything?
Here's the thing. They're gonna ask you about the proteins.
Okay.
They'll say like which of the following is the thin filament, in that responsible for muscle contracting. The thin filament's the actin. It's got the binding sites, right?
Mhm.
They might say, what's the thick filament? That's the myosin. Or they'll say like, what is the proposed theory for how muscles contract? That's the sliding filament theory, right?
Okay.
You might get questions like that. Are they going to ask you like, to describe this whole process like I just did? No, definitely not.
Okay.
But when I give you this like big, hopefully we've painted a really clear picture in your head and you're seeing now in your head like all this like sliding stuff, it helps you understand like when a muscle's tight and it's got a knot, right? Let's take a look at what that kind of looks like. Look at the fibers.
Yeah.
Right? The actin and myosin proteins are contracted here and here, right? So that's what a knot actually is, right? Because it's all these like bundlings. So hopefully we've painted a picture. So now we know we can get in there and just work that knot out, right? And we're going to go with the grain of the muscle in order to make sure that that happens, because we're trying to pull the myosin and actin proteins apart. Or maybe I just pull it end to end. I'm just trying to pull them and get them to relax. Or maybe I'm just trying to get a signal from my nervous system to get to these freaking stubborn fibers here that's going to say, hey, shut up.
Yeah.
Like, because that's what they're doing. They're just, they're going, ah, and they're just like, like, and I'm just going to try to get a signal from my brain that says, hey, let go, So that's why we're looking at all this. Again, it gives you a deeper, richer understanding rather than just like, oh, the book says I should stretch for 30 seconds.
Yeah. No, that's perfect. I love it.
All right. So we got 2 more things to get through today. And this stuff is actually a little bit more of the language we're actually going to use. So muscle fiber types, you are going to have 2 different types of muscle fibers. And what I mean by that is like, they're just going to have slightly different characteristics. And so we're going to name them after the character. It's like having green fibers and yellow fibers. You know what I mean? They have, what are their, it's like the characteristic is that one's green and that one's yellow. They look different. So we're going to name them slightly different based on their characteristics. Okay. So some muscle cells are going to look a little bit different from other muscle cells based on characteristics. So muscles vary in their chemical makeup, their mechanical makeup, and so it gives them specific characteristics. Your type 1 muscle fibers, the characteristics that you're going to see in type 1 fibers, these are sometimes referred to as red fibers because one of their characteristics is they have a ton of blood flow going to them. So they get stained red. And so red fibers tend to be like very small in size because these are your endurance fibers. Okay, these are often associated with stabilization. These are often associated with posture. These muscles that can contract for a really long time. I've been using my jaw nonstop today, right? Those are very much type 1 fibers that are just going on, and they're just there and they're just not stopping. If you were to take like a tennis ball, right, and just squeeze it over and over and over and over and over again, and just do a whole bunch of reps, right? Getting a lot of repetitions in right now, right? I am mostly using endurance fibers to do that. If I go for a run, I'm mostly using endurance fibers to do that. Those are your type 1 fibers. Now, they're endurance fibers. They're not very big. There's a very big difference between like someone who uses those fibers all the time. Let's say like a marathoner, right? A marathoner, they're gonna be a long, skinny person with lots of like type 1 fibers that they developed over time, right? Lots of oxygen being delivered, muscles are smaller, they're very good at long-term contractions, they're very good at producing energy over a long duration. But, are you gonna choose like, this guy to set the world deadlifting record? Probably not. So we have other types of fibers as well throughout our body. These are called your type 2 fibers. So your type 2 fibers are often called your white fibers, and they tend to be much, much, much larger in size. And the reason that we call them white fibers is they don't get stained quite a deep color of red. They aren't quite as red as your red fibers because they don't get as much blood flow. So that means that they are going to be much bigger in size. So without that blood flow, like they're not going to be able to produce as much energy. So like blood is what delivers oxygen, right? Oxygen is how we produce energy. That's why type 1 fibers are our endurance fibers, whereas type 2 fibers, they can create like a big burst of energy that's like really intense. It's like, you know, and it like throws something super, super far. But it can only do it for a very short duration of time because it runs out of oxygen and it can't really keep going. So those are your fast twitch fibers. And that's what we're going to see in like a sprinter. Like you look at a sprinter, they just, okay, Google. That's so dumb. You look at like a sprinter or like a football player, right? There's a really big difference here, right? There's a marathon type 1 body, right? There's a sprinter type 2 body, right? Both of those athletes are probably incredible athletes at the top of their game, you know? But their bodies do look very, very different. Even like, you know, Usain Bolt's like a pretty thin dude, but like you can see there's a big difference between him and like Eliud Kipchoge. This is the world's greatest marathon runner right here. This is the fastest man on the planet over a long distance. This is the fastest man on the planet over a short distance. And they just look a little bit different, right? And that is where we are seeing a slight difference in their muscle fiber makeup because of the way they've trained. You know, they've trained to develop those different muscle fibers. So type 2 fibers are your fast twitch fibers, right? They're larger in size. They don't have as much mitochondria, so they can't produce short-term energy, but they have more sarcoplasm. That's part of why they're bigger. Therefore, they can produce lots of really fast energy, but it runs out very quickly. They don't have as much blood flow, so they can't produce energy consistently, but they have some blood flow, which allows them big burst of energy, right? Now, we are going to split type 2 fibers into like a little subcategory here. We've also got what are called type 2X fibers for extreme and type 2A fibers. So type 2A fibers have a little bit more oxidative ability. They tend to have like some more enzymes and they get a little bit more oxygen. They're kind of like they're like like type 1.5 almost, right? Whereas type 2X is like true fast twitch, low oxidative, no enzymes, just a lot of proteins, right? They are there to contract once, maybe twice, maybe 5 times, you know, like that's like just true explosive, right? So now we're starting to see the difference between maybe like a runner, it's like a type 1 athlete, and then like a bodybuilder, which would be kind of like a type 2 athlete, and a power lifter or an Olympic weightlifter, which is like a type 2X athlete, right? Think about the sport of Olympic weightlifting, right? The job, that whole sport, right? What's the job? It's to lift a barbell overhead once. Sport over, you know, like that's type 2X, right? So are they training and doing like 20 minutes of cardio in the gym? No, it's not related to their sport, right? Are they doing 20 repetitions of an exercise? No, it's not related to their sport, right? They are lifting a heavy barbell overhead one time and then they're done, right? Now look at a football player, right? Let's say, you know, that I would argue that that's also very type 2X, although certain parts of it are very type 2A. You think about like a wide receiver, right? You know, they're sprinting down the field, and then like they're taking a shot, right? And then they're doing it again and again and again. Maybe they're doing it like 4 times in a row, but like, you know, that's very much like they need a little bit of endurance, but like at the same time, they're trying to beat their opponent. So they're trying to be very explosive. So they're probably like a type 2A athlete, maybe a little type 2X training in there, just out the gate, right off the gun, like the really hard, like, you know, big sprint in the beginning, and then like they kind of slow down a little bit. So they use their type 2X fibers and they get into their type 2A fibers, right? That's oftentimes when we think of sports guys, we are talking about type 2A fibers. Most sports rely on those. And when I say most sports, I mean like field sports, basketball, right? Like LeBron is not going to be as strong as a power lifter, but he's going to be way stronger than like a marathon runner, you know? And that's kind of the difference that we see there. So those are various, you know, different types of like muscle fibers out there. This is why, like, you know, like DK Metcalf, like, love the guy. you know, but he wanted to like try to be like, he wanted to like qualify for the Olympics. And so, because he had like a, you know, he's at a really great 40 time. Like his 40-yard dash was great. But, you know, the Olympics doesn't have a 40-yard event. It has a 100-yard and it has a 400-yard, right? And so when he tried to like go that extra 60 yards afterwards, you know, that's the difference between like those two types of athletes, right? He's got to be able to play, you know, over and over and over. He's got to do like one after the next when it comes to like doing all these plays in a sport that requires it to happen multiple times. So he's very much not training for just explosiveness, even though the distance that like, you know, he's training for is still relatively short. There's just a difference between the way that he has trained his whole life to become like an elite level athlete versus like an Olympic sprinter and the way that they've trained to become an elite level athlete. They're just different sports, you know? So that's the difference between our type 1 and type 2 fibers. And so you can see all the characteristics kind of laid out here. And obviously you can see those characteristics a little bit when you look at things. Any questions on that, guys? Any questions on muscle fiber types? Alrighty. Well, lastly, let's take a look at how we're going to label out our muscles to understand movement. So here's the thing. I'm going to talk about your pelvis tilting. I'm going to talk about, you know, your shoulders rounding. We're going to talk about all these like movements that are occurring, which means we need to understand which muscles are controlling those movements. Okay. So we got to have a very clear way to label which muscle is doing what action at what time. Let's go back to the QL as an example, right? Remember earlier we talked about the quadratus lumborum, right? The QL, right? It's in charge of this and this. And they're opposites, right? You know, I'm dancing on camera. This is a low point in my career. Now, so we talk about like those two different muscles, right? Controlling my pelvis, like back here, moving me, you know, this way or this way, right? Both of those muscles do it. But if I go to my right, that's my right QL contracting. If I go to my left, that's my left QL contracting. So they are opposite of one another because of where they're situated on the body, right? This is the opposite of this. Left is the opposite of right, okay? So we need to have a way to kind of label. If I'm trying to figure out, like if I've got a client who's shifting with their pelvis, right? To the, you know, their right side is pulling up, or maybe they're tilting, you know, down like this, right? If I know that it's their right side, then I know that the right one, which is the one that's contracting, is tight, which means the left one is weak, right? So what I need to do is I need to stretch the right and strengthen the left. And so I have to have an understanding of how I'm going to label this in order to understand these joint actions. Your bicep contracts when your tricep relaxes. Your tricep contracts when your bicep relaxes. They are antagonists to one another. So your muscles do provide a variety of different functions. We're going to categorize those into 4 primary jobs. And one thing to keep in mind, this is a direct quote from your trail guide book. Keep in mind that in the course of one simple movement, a muscle's role can change. It can go from beginning as the main muscle, which is we call that the agonist, and then quickly switch hats and become like a fixator or a synergist. It can suddenly become like a helper muscle. So a muscle, any muscle can be the main muscle or the opposite muscle or a helper muscle. It just depends on what the movement is that's happening. So for instance, here's our 4 terms. Agonist, right? An agonist is the muscle that's primarily responsible for moving a joint. So, or responsible for like an action. So for instance, these are my hip flexors, right? This is my hip flexor complex right here. So they are primarily responsible for hip flexion, right? When I perform this movement here, where my femur comes up like this, my hip flexors are primarily responsible for that. They are the agonists. Meanwhile, on the complete opposite side of my body, my gluteus maximus back here, that is the antagonist. because it has to relax when I pull my thigh up like that. If I go like this muscle has to relax here. Why does it have to relax? Well, it's because it's in charge of hip extension, which is exactly the opposite of hip flexion. So my glute is in charge of that. Hip flexors, glute. So they are opposites of one another. So the agonist is the main muscle responsible for a movement. So it depends on like what I'm talking about. If I'm talking about hip flexion, the agonist is the hip flexor complex. Now, if I'm talking about hip extension, the antagonist is the hip flexor complex, right? Like they would flip roles. So when I'm talking about elbow flexion, my bicep is the agonist, my tricep is the antagonist. When I'm talking about elbow extension, my tricep is the agonist, my bicep is the antagonist. So it switches roles, right? I mean, not even roles. It's switching, like, you know, it's all contextual based on how I'm talking. So the agonist is the prime mover for a joint. Your gluteus maximus, for instance, is the agonist for hip extension. So if you're squatting, the main muscle is your gluteus maximus. Here's how I like to think about this. Yeah, question.
Brad, can you repeat it one more time? That you just explained the agonist and antagonist.
Yeah. So the
extension and
yeah. So the agonist is the main muscle for a job. So if the job I'm talking about is a squat, right? So let's say like I'm here and I go down and I come up, I squat, right? The agonist, the main muscle that did that was my glute, because think about it, I'm going from here where I'm in a hip flexed position, right? I'm up like this, and then I'm going to a hip extended position, right? I'm extending my hip, which is bringing me back up. That's my glute that's doing that, right? So my glute is the agonist. Meanwhile, the antagonist is on the other side of the joint. That's my hip flexors. So flexion's the opposite of extension. The muscle that does flexion is opposite of the muscle that does extension. Those muscles are antagonists. Here's the analogy I like to use. It's Batman versus the Joker, right? Like Batman's the main guy. He's the main character responsible for like keeping Gotham safe. That's the action, right? The antagonist is the Joker. Exactly totally the opposite of Batman, right? Now, there are muscles that are there to help. There are muscles that work with the prime mover, right? The muscles that work with Batman. So these are what we call synergist muscles. So for instance, when I was coming out of my squat, there's another muscle that also does a little bit of hip extension. It doesn't do it super well, but it does some hip extension. That's my hamstrings. So my hamstrings back here, when I'm coming out of that squat, my hamstrings also are contracted. They do a little bit of hip extension at the same time. So they're an assister muscle to my gluteus maximus. That is a synergist. Okay. So think of that as like Robin, you know, Robin is Batman's sidekick. He's there to help. Both of them are working against the antagonist. They're both working against the Joker, right? Now, obviously, this analogy breaks down if I immediately go in the other direction, you know, like suddenly, you know, like for instance, like if I'm doing a pull-up, right? Let's say I'm doing a pull-up here. If I'm going like this, right, my agonist is going to be my lats, and a synergist is going to be like my posterior deltoid, or you know, my biceps are there to help, right? So the main muscle, Batman, is my lats, and my helper muscle, Robin, is going to be my biceps, right? But if I were doing a shoulder press, which is practically the same thing, suddenly everything flips around, right? Now my agonist is not my lats. Now my lats are an antagonist. My agonist is the deltoid, and not my biceps, my triceps, right? So everything can flip around depending on what it is you're talking about. And that's why we have all of our terminology that we're going to get into tomorrow, well, not tomorrow, Monday and Tuesday. It's going to pair with this. But for now, you got four main categories. You've got the agonist, which is the main muscle responsible for a job. The antagonist, which is exactly the opposite of that. A synergist, which is working with that muscle for the job, right? Agonists and synergists work together to move. And then there are stabilizers as well, which I haven't talked too much about. Stabilizers are there to support the body while you're creating those movements. So for instance, I might have like a stabilizer muscle in my elbow while I'm doing this, or my rotator cuff or my core, right? My abdominal muscles, which are holding my spine in place. Those are stabilizers, right? Think of it as like Alfred the Butler. You know, he's there to provide emotional stability for Batman. So that is sort of what we're talking about when we're talking about movement. So when we start getting into, you know, looking at altered posture, right? When we see altered posture, again, let's see, we have a client with an anterior pelvic tilt. It's a good example of an alteration in posture, right? You know, we see this exaggerated low back arch, right? I know that this agonist muscle over here, the hip flexor complex, is overly tight. So I need to stretch that out. The antagonist muscle to that would be the gluteus maximus, which is weak. And then the other antagonist to that would be the hamstrings, because that's working with the gluteus maximus. So both of these muscles need to be strengthened, and this muscle needs to be stretched. And I'm going to know that because I know that this is antagonistic to this and this. And then stabilizers, and I need to work on that as well. But does that make sense? Does that start to sort of paint a picture of where we're headed over the next, you know, couple days?
Yes.
Yep.
Cool. And that's why we're looking at this. That's why we use this terminology. It's like, we just want you guys to get familiar with like agonist antagonist relationships. Like, you know, do you guys ever play, I'm going to use this analogy again later, actually. Do you guys ever play catchphrase? It's the game where, you know, you're not allowed to say the word, or heads up is another one that you play. But it's like, you're not allowed to say the word that you want to get your partner to say, but you're allowed to say whatever else you want.
Yes.
So like, maybe I would say something, you know, I would lead you there in a classic example, by being like, oh, he's a DC Comics character, you know, he's dark and brooding, you know, and be like, oh yeah, Batman, right? That could get you there. Sure, it could. But to me, my favorite strategy whenever I'm playing that game is I just say the exact opposite. I'm like, it's the opposite of the Joker. And so I go, oh, Batman. Right? That's a much easier way to kind of get there. That's when you can kind of start to think that way, think of like what it is you're trying to think of and the reverse. That really helps with you understanding posture. And it really helps with you understanding like what muscles to strengthen and how to flip over to the other side of the joint and what muscles to stretch. And it all is because like some of the fibers are slid together. Meanwhile, the other fibers are all pulled apart. So that's why we looked at the microanatomy today. You know, you want to picture these working parts. We're going to see this come back again on Monday.
Is it the agonist is always the muscle that you have to stretch?
It would technically depend on how it is you're framing it, like how you're talking about it. You know, like if I were saying excessive hip flexion, well, I'm talking about hip flexion now. So yeah, the agonist is the one I need to stretch. But if I were talking about like, oh, you have a lack of hip extension, well now, because I'm talking about the other side of the joint, I'd say you need to stretch the antagonist to hip extension. Which would be, you know, like, so it's all contextual based on how you're describing it.
Okay.
I will, that if that doesn't feel clear right now, it will on Tuesday. It'll feel clear when it comes back around. But for now, just get those 4 categories and terms memorized.
Okay, thank you.
Yeah. All right, and that'll do it, guys. We made it to the weekend.
Nice.
Cool.
Ready to hit the gym now.
Hey, I love that. Yeah, me too, actually. I'm gonna go get some food and then work out around noon today.
Yep.
All right, let's